Bismillahirrahmanirrahim. Let's continue with the paper 4.2 which we started earlier in a previous video, and we've done question 1, 2 and 3 in that, and we continue with the question 4, 5 and hopefully the question 6. Now in question 4, I'm just going to read this to you. Adult Drosophila fruit flies feed on yeast on the surface of ripe fruits. Female flies lay their eggs inside the fruit, and the larvae feed inside the fruit before developing into adults. The tree Morinda produces fruits called noni fruits. Now noni fruits contain a toxin and are avoided by nearly all Drosophila species. So Drosophila was the fruit fly, and this is a fruit, so including Drosophila jacoba in mainland Africa. One population of D. jacoba lives on the island of Mutation off the coast of Africa. The population of D. jacoba on Mutation has specialized to feed on noni fruit. Flies of this population produce enzymes. So they must have the gene for that enzyme that gives them resistance to the toxin on noni fruit. So they are not going to be killed by this toxin, and they are resistant to it. Now adults are able to feed on yeast on the fruit surface, and their eggs and larvae can develop and grow inside the fruit. Scientists have used bioinformatics to estimate that the unique D. jacoba population on Mutation originated in the last 30,000 years. Describe how a population of Drosophila can develop resistance to noni fruit toxin. Now please remember, whenever there is a question on this selection, mutation, resistance, there are some keywords that we have to be using, and those keywords are all in the context of mutation, advantageous allele, and then passed on to offspring, and then of course the entire population becoming resistant. Now you can see this was a 4 marks question, and there are about 7 mark scheme points. Now these mark scheme points you need to sort of memorize, because you can write these in any question which is going to be testing this portion of the syllabus, which is selection and evolution. So now the first mark scheme point, as is very obvious, is it's a spontaneous mutation. It just happens. You must remember, flies reproduce very fast, and if 1 or 2 mutate, then it's going to be passed on to generations, because they lay so many eggs, and then your eggs develop into adults and all that. So it's a spontaneous mutation which just occurs. That's the first mark scheme point. Then produces an allele coding for that enzyme, which we just talked about in the question. Now the toxin is the selective pressure. Then flies with allele have selective advantage. A very typical biological English, you need to remember this. These flies survive and reproduce, pass on this advantageous allele to the offsprings, and this allele increases in frequency. So there were a number of mark scheme points, and if you gave me any 4 of these, you'd probably get your 4 out of 4. Now the B part of the question is a little difficult, because it is now testing your knowledge of this chapter. It says explain why living on an island increases the likelihood of the non-toxin resistant population of D. jacoba on Mutation becoming a separate species to the mainland population of D. jacoba. So why on this, when they live on an island, this increases the likelihood that the non-toxin resistant becomes a separate species. So then of course you must know the definition of species, and then you must know why are we saying that it's going to become a different species. Now the question said very clearly that this was on an island, and then there was the mainland population. So there was the clue for the answer, that this is separated by a geographical barrier. And this of course is called allopatric speciation. This is called allopatric speciation. That's a name that we have studied in this chapter, is allopatric speciation, when a species arises because of a geographical barrier, and then they're not allowed, they mix, and so their mating behavior changes, or the timing of flowering changes, or something else changes which makes them a separate species and they cannot interbreed to produce fertile offsprings. Now there could be different selection pressures. Mainland could have different weather conditions or humidity or wind or something like that, which is of course it would be the selection pressure. Then these are reproductively isolated, and this is pre-zygotic isolation, because even before the zygote is formed, they are separated and they do not mate and they do not interbreed. So they do not breed to produce fertile offsprings. Then genetic drift, when a small number is separated from the main, and then of course it changes the allele frequency, and this is of course more pronounced in a small population. So you've got to understand all these points, and you've got to understand that they did give you a clue that there was the mainland population and then this was on another island. So there was a lot of geographical barrier between the two, and how you would figure out. So there's always a clue in the question when you're doing a paper 4 question. So you must take time to read the question and understand it, and then figure out what the possible answer could be. Now the part C of the question was very straightforward, wasn't a very difficult question. It just says suggest, and the suggests are always the one which are going to differentiate between the A star students and the A students. So suggest, either you know it or you don't know it. So if you know it, do it. If you don't know it, just leave it. Suggests how scientists can use bioinformatics to estimate the length of time that the population of D. yucuba had been living on Mayotte. Now we're using bioinformatics to really find out how long have these been living here on this island or on this place which they have mentioned in the question. Now, as you all know, bioinformatics just collects data, and these are these huge databases where we have all this information stored. So we could obtain the DNA sequence from the database. We could find out, when we know the DNA sequence, we can find out the amino acid sequence from the database. Then we can compare the sequences of two populations. Then we can compare or calculate the percentage of bases or the amino acids that have changed between the two populations. Or we could analyze the mitochondrial DNA. And then of course we could use the molecular clock and also the mutation rate to estimate the time. So we had 6 mark scheme points, but we were only supposed to give 3. So if you gave me the first 3, well, you got your 3 out of 3. So you have to understand, number 1, obtain DNA sequence from database. This of course will help you to also estimate the amino acid sequence from the database. You could either do that or this, or if you wrote both of them, that would be correct as well. And then you compare the sequences of the two populations. And then you calculate the percentage of bases that have changed, or you could use the mitochondrial DNA and look at the nucleotide or the DNA sequence in that. And then of course this would help you to find out from the molecular clock the mutation rate. Then we come to question 5a. A severe reduction of blood flow to the brain. Now please also remember this, if this question is question 1a, then it has part 1 and it has part 2, and you have to see if there is anything. Oh yeah, there's a part 3 as well. So you see, all this is interrelated to the information given to you in the beginning. So please understand this, that if it's question 5a, so then read the A part very carefully and then start answering part 1, part 2 and part 3. So let's look at the question. A severe reduction of blood flow to the brain causes cells to die. Reduction of blood flow. This is called a stroke. Now the after effects of a stroke can range from recovery to permanent brain damage and death. So either you recover or you have permanent brain damage or it's death. A new emergency gene therapy treatment for people who are at risk of brain damage from a stroke was tested in mice. The human granulocyte colony stimulating factor AGCSF is a protein that stimulates the production of stem cells in the bone marrow. First information given to you. Second information given to you, mRNA coding for the AGCSF was obtained and used to make cDNA. Fine, so mRNA, we have mRNA and then we're going to make what? We're going to make cDNA, which is copy DNA, which of course does not have any introns. The cDNA was inserted, and we got the cDNA was inserted into an adeno-associated virus, AAV, vector, and given in eye drops to mice just after they experienced a stroke. So we have this virus, and we've somehow, this is a DNA virus, and then we put in the cDNA, which I'm putting in green, and we've added this to this viral DNA, and then we're going to give it as eye drops to mice after they experience a stroke. Now let's look at, first of all, we've understood the question. Now let's look at what are we going to answer. What is meant by gene therapy? So this is a direct question. And then it says, describe the role of reverse transcriptase and DNA polymerase in making cDNA for AGCSF. Again, a very direct question. And then it says the AAV vector used was unable to replicate itself within the target cells. Suggest why the researchers chose a vector that could not replicate. So we have to read the whole question and then we start answering it. Now in part 1, what is meant by gene therapy? It's a very direct question. You have to say what was gene therapy. Gene therapy is a form of therapies to treat a disease caused by a faulty allele. So the body has a faulty allele. What we're going to do is we're going to use a vector to give the correct allele so that the correct protein is made. So number 1, to treat a disease caused by a faulty allele and deliver the allele to the target cells. And then we did this in a disease like cystic fibrosis, which has a chloride ion channel which is defective. So if we have got the normal allele for that, then we will somehow make it reach the target cells so that the normal protein can be made and not the defective protein which is being made because of the faulty allele or the wrong allele, which is resulting in the formation of the protein either being not the right shape or not having the right active site or not having the right receptor or not being able to function as a channel protein which is actively transporting chloride ions out of the cell. Then it says, describe the role of reverse transcriptase and DNA polymerase in making cDNA for. So first of all, uses mRNA to make the ssDNA. ssDNA is single-stranded DNA and makes DNA double-stranded. So I've just shown you an mRNA and now I'm going to show you the single-stranded DNA it's going to make. So A will have a T here, T. Please remember now this is the single-stranded DNA. So then we have A here, A here, A here, G here, G here, G here, and a T, and a T, and a T. Please remember T in DNA, mRNA doesn't have a T. And then it's going to make the double-stranded. So the DNA polymerase is now going to, so we're going to add an A here. And then this is a T, and then a C, 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 and an A. Now, DNA polymerase is going to make the sugar phosphate backbone. So here now we have cDNA, but this is a double-stranded DNA. This was initially a single-stranded DNA, and this is the mRNA from which it was made. Then part 3 of the question, the AAV vector was used was to unable to replicate itself within the target cell. Suggest why the researchers chose a vector that could not replicate. Now, basically why we did not want it to replicate is because we didn't want copies of it to be made, number 1, to prevent the virus from spreading and to limit the immune response, or you could have said limit the side effects, or you could have said limit maybe production of cancer could have started, or to also not have the illness becoming more profound or causing the infection to spread. So we didn't want the vector to replicate. So we didn't want it to spread in the body. Just like any virus, we're just going to use the host cell and it's going to replicate inside it. So we didn't want it to replicate because that would maybe harm the, produce side effects like resulting in a cancer or resulting in some severe side effects, some allergies, some reactions. Now let's look at the B part of the question. So we finished the A, 1, 2, 3. Now we are on the B part of the question. Now you need to study this very carefully because now they're going to ask you something about what the information they're giving you. So study was carried out to investigate the effect of gene therapy described in A. 4 groups of mice were used. So they're telling you the information is that 4 groups of mice were used. Group A mice had a stroke. They received eye drops containing AAV vector carrying cDNA for the AGCF once only. Group B had a stroke. So group A and B both had a stroke. They received eye drops containing AAV vector carrying cDNA for 4 times. So here there is once and then it's 4 times. So you please be very clear about this was given once in group A and 4 times in group B. Now group C had a stroke, so they had a stroke as well. So all of them had a stroke. All of them had a stroke. Only this one did not have a stroke. So D did not have a stroke. So this is the D group. So group C had a stroke. They received eye drops containing AAV vector carrying the GFP gene coding for green fluorescent protein instead of the cDNA for AGCF, and this they only got it once. So group C mice had a stroke. They received the eye drops containing the AAV vector carrying the GFP gene, which is coding for a green fluorescent protein instead of the cDNA. So they did get the vector. So the 3 of them had a stroke. This one had a stroke, this one had a stroke, and this one, group D, did not have a stroke, and they were not given any eye drops. Now, first question. Explain why the mice in group C were used in this study. And group C had a stroke, they received eye drops, but they had a gene which codes for a green fluorescent protein instead of the cDNA. Now, if you look at this first part of it, it says explain why the mice in group C were used. Basically, they were used as a control because you see they had a gene coding for a green fluorescent protein, which is of course the identifying gene because it fluoresces, but it didn't have the actual cDNA. You see, all of the others had the actual cDNA, but this one did not have the cDNA for the AGCSF. So number 1, it was a control. Number 2, we wanted to compare the effect of no gene therapy with gene therapy. Did it really work? Did it work in group A and group B, and then it didn't work in group C because they didn't have the gene? And then to see where the gene goes, so that if we had a fluorescent marker, then we would know where it was going, which cells would show that it had really reached there. And then to see the effect of the virus alone. The virus alone, did it do the same effect? If it did, that means the gene with it was just going to do because of the virus. It wasn't because of the gene for CSF which we had given in group A. You see, group A and group B had the cDNA for AGCSF. One had it once, one had it 4 times. This one did not have this, so there was no gene for CSF, the cDNA for AGCSF in this one. So explain why the mice in group C were used in the study. So you need to be really understanding this, and so that you are able to answer this question. Then in the part 2 of the question, it says explain why the mice in group D were used in the study. Now group D, if you look at this group D here, they did not have a stroke and they were not given any eye drops. Now why would we do that? To compare the results of the mice without the stroke to the mice with the stroke. So we do a comparison. How did they fare? And then to establish a baseline figure, which is a reference point, and to make the study valid. If you look at this point, why the mice in group D were used in the study, we says to make the study valid. So if we do a comparison that the ones with the stroke had this issue and the ones without a stroke, and how did they fare? So then we could study or so we could use it as a reference point. That was another point that they gave you. To make a study valid, 1 point, to make it to act as a reference point, and to compare the results to mice with a stroke to mice without a stroke. Then the part C of the question is checking whether we've understood this whole study and we can answer the questions on it. Table 5.1 summarizes some results from the study and shows the percentage of mice surviving, the percentage of brain volume occupied by fluid-filled space, and the score on a behavioral test in which normal mice score 0.5 and brain damaged mice score nearer to 1.0. Normal mice 0.5, brain damaged score nearer to 1.0. So please read the question carefully and make a point to underline and understand. Now we have a comparison, we have a table which we need to go through. Mouse treatment group A, 63% survived. This is a percentage of mice surviving. In B, 100% survived. In C, 25% survived. And in D, 100% survived. Now percentage of brain filled with fluid filled, 3.6% had brain occupied by fluid filled space. Here we had 3%, here we had 5.2%, and here we had 3%. Then behavioral test, we said the normal mice would score 0.5. Now here we have a 0.67, so this is brain damaged, this is brain damaged, this is brain damaged, and this is of course the one which will be the normal mice. So please understand, score 0.5 was for normal mice and the brain damage scored something nearer to 1.0. Now let's look at the question. It says use the results in table 5.1 to evaluate the benefits of gene therapy treatment with AAV vector carrying the gene for AGCS for people who have a stroke. So you now need to evaluate. So you need to look at this data and then give us your idea or your thoughts about it. That was this helpful? Should we give this to people who have had a stroke? So evaluate the benefits of gene therapy treatment and carrying for people who... So this is for people. Now remember, we didn't do this on people. We did this on mice. So now think before you answer this question. Now let's look at this table again and understand. You see, for the group of A group, we only gave them the eye drops once, but the B group, we gave them 4 times. And their percentage survival is 100%. So this is increasing the rate of survival. The C ones did not get it, their GFP, so only 25% survived. They only got the GFP, they didn't even get the actual cDNA. And these one had no stroke, so 100% survived. Well, they would have all survived because they didn't have any stroke and no drops were given to them. So the first point is that increases survival. And the next point is, now when you look at it, this was 3.6 and the percentage of brain occupied is now 3.0. So this reduces the percentage of brain occupied by fluid filled space. This reduces the percentage of brain occupied by fluid filled space from 3.61 to 3.0. So this was another point that you could have given me. So it reduces the fluid filled space, which I've just explained to you here with 3.6 and 3.0. Now another factor which was helping, look, it says 0.67 and then 0.67. Now if you remember, 0.5 was the normal one. So these are, now the behavioral test also tells us that improves or lowers the behavioral test. So it's close to normal. Now in this one it was 0.9, this is close to 1. And the D group is of course 0.5. So these were close to 0.5. I'm not saying they are. So 0.67 is close. So improves the behavioral test score. So the third point would be improves behavioral test scores. And I've just explained that to you. So as you look at it, I mean, we have all these points which we have picked up from this table. And increases survival, reduces fluid filled space, improves behavioral test score, eye drops 4 times better than 1. If effective with mice, likely to be effective with humans. Humans and mice very similar. We have we do a lot of drug testing on mice. But then of course this is also not very distinct because we've only tested on mice, so we don't know how it's going to react, how humans are going to react to it. So you can write an adverse remark on that as well. And then of course you can say treatment is non-invasive. You don't have to give an injection, you just have to give eye drops and it is quick. So there were all these different points and you had to give me any 4, but there are many points which you could have given me. There's 1, 2, 3, 4, 5, 6, 7 points. So 7 points you could have given me, and then you could have given me a comparative data code. And the data code is to support either one of the marking point 1 or 2 or 3. And this is the process data which you could have given me. And I'll just show you an example of it. See, what was the marking point 1? It was increases survival. Now you could have compared 25% with 63%. Or you could have compared 25% with 100%. Or you could have said 63% versus 100%. So this was actually telling us about the survival. So any of these 3 comparisons, 25 versus 63, this one, then 25 versus 100, this one, you could have talked about this one, or you could have said is 63% versus the 100%. So these were the, if you gave me any one of these figures, well, that is to, you say, the data, comparative data code to support the one of the marking point 1. Now for the marking point 2, which is here, reduces fluid filled space. Now which comparison would we do? Now if you look at it, this is the one we're going to compare. Now in this one, which are we going to compare? We're going to say 5.2% and 3.6%. So you're going to quote this, or you're going to say 5.2% or 3.2. So you could do this one. If you could compare this and this, that was fine as well. Or you could compare the 3.6 and the 3. So you could compare this and this. So this is how you would give me, that's called a data code to support marking scheme point 2, in which we say reduces fluid filled space. Now for the third, I mean, this is only going to get you 1 mark, but I'm giving you the different options because this is how I'm training you to answer these questions. Now if you look at the marking point 3, it says improves behavioral test score. Now here, what are we going to compare? Here we are going to compare 0.90 versus 0.67. You see, 0.90 did not get any medicine. They were not given any medicine. They were given the GFP. And 0.67 is the one which was given 4 times. Right? So you compare this with that and you see that, okay, improves behavioral, it became more towards 0.5, which was of course the normal one. So normal my score was 0.5. So this is what you had to remember, and you had to look at this part of the question. Normal mice score 0.5 and the brain damaged one had a score of 1.0. So here there was 1.0 score and here it was a 0.5 score. So now which one is near 0.5? So 0.90 went up to 0.67, which is nearer 0.5. So this was the marking scheme point which was then being tested for the third. This is improves behavioral test scores. So this is what we had to answer for this part of the question. Now this was a 14 marks question, and this was quite a difficult question as far as I'm concerned. And what we need to learn from these sort of questions is, number one, you read the question very carefully, and then you think of what possibly could be the answers. And maybe if you're struggling with the answers, then you need to go on to the next question and then come back to it later on if you have time. So whenever you have a difficult question, remember, you need to come back to it later on. And thank you for now, and I'll continue this in another video.